it is my presentation about the etiology and pathophysiology of Ewing's sarcoma. Initially, let me to clarify what carcinogenesis means, what apoptosis means, and what is the correlation between apoptosis and the specific enzyme activity called telomerase. Carcinogenesis is a formation of malignant cells resulting in the loss of balance between normal cell proliferation and so-called apoptosis or programmed cell death. There's a strong correlation between apoptosis and a specific enzyme activity called the telomerase, which was actually discovered by Elizabeth Blackburn, an Australian-American scientist who received the Nobel Prize in 2009. Telomerase activity is very high in malignant cells, stem cells, and gametes. It's actually produce a guanine-rich tail at the end of the chromosome, which is actually protects the chromosome to fuse together. Telomerase activity is getting less and less when we age, and naturally also and regularly it produce apoptosis as well. Carcinogenesis can be caused by environmental factors, any contamination in the environment, viral infections such as, for example, cervical cancer caused by human papillomavirus, accidental DNA mutations such as single nucleotide polymorphism or SNPs. Quite often it can cause breast and skin cancers, and in several other cases which are unknown. Ewing sarcoma was first described by James Ewing, named after him, around the 1920s. Ewing sarcoma is a malignant growth in the bones and surrounding tissues. It is one of the most common tumors in childhood. Quite often it happens between age 10 and 15. Later it was established that Ewing sarcoma is not a unique tumor. Morphologically, it is a heterogeneous tumors that are characterized by so-called non-random chromosomal translocations. So we called it a Ewing sarcoma family of tumors, ESFT, which includes Ewing sarcoma and the so-called peripheral primitive neuroectodermal tumors, which is actually accepted by the World Health Organization. Why? Because they have very similar histological profile and similar gene prearrangement, which is actually presented in this figure. During the development of Ewing's sarcoma, a non-random chromosomal rearrangement takes place, which means that gene translocation of the Ewing sarcoma gene, which practically means swapping the pieces of DNA between two genes, probably between chromosome 22 and 11. It's not just the gene will be translocated, because it looks like it's a linked gene, and the translocation happens together with the so-called transcription factor, and it's probably responsible and happens about 95% of the development of Ewing's sarcoma. Why this translocation takes place, we don't know. Can be inherited, we don't know. This figure just shows you, refresh your memory about the chromosomes. So the short arm is the P arm, the long arm is the Q arm, and the stripes are the chromatids, and in the middle you can see the centromere. Actually it represents where the two maternal and paternal chromosome joins together. To precisely diagnose Ewing sarcoma, it's quite a few steps involved. Early signs shows persistent pain of the patient, swelling, and some function impairments, especially if it happens in the ribs or the femur. Radiological findings normally followed by some specific examination, such as histological examination, immunochemistry, and molecular analysis. Histologically, Ewing's sarcoma belongs to the so-called small round cell tumor group. As you can see on this photo, they stained as eosinophilic, small cells with fairly high ucrol to cytoplasmic ratio. Immunohistochemical staining can be accomplished using antibodies when we attach antibody to the target antigen and the secondary antibody, which is normally labeled by an enzyme, for example, a horseradish peroxide, and adding as a substrate, we can detect color reaction. Molecular analysis means the detection of specific gene sequences resulting from the translocation from chromosome 22 to 11, together with the transcriptase gene sequence as well. Recently discovered several novel biomarkers to help detect Ewing sarcoma. There can be diagnostic tools, for example, detection of reciprocal translocation between chromosome 22 and 11, can be prognostic, looking at the genomic stability of the rearrangement, and can be predictive, for example, to carry out microRNA deregulation analysis. MicroRNA are non-coding RNA sequence, and they actually regulating gene expression. How we treat Ewing's sarcoma? It cannot be cured, but we can do surgery. We can do combination of chemotherapy and radiation therapy, and some not new, but fairly recent targeted therapy, for example, using monoclonal antibodies. Concluding my presentation, let me give you some references or links which could be useful to get a better insight of this cancer. Thank you very much for your attention.